Thanks, Connie, and thanks to everyone for tuning in. I'm Cecil Yule, and I run the Genomic Applications team at ONT. Every year at NCM, I give a talk, and I'm known to bring in people from the team to talk about their projects. This year, I'm bringing in Phil James, who runs our clinical applications team, and he's going to talk about another project in about 10 minutes. The Genomic Applications team is spread out all over the world, but we have labs in Oxford, New York, and San Francisco. We do a lot of different things, and the project that I'll talk about today is from our showcasing team, mostly out of the US. We try to showcase the ONT technology in the best possible way. And the project I'm going to talk about today is extrachromosomal DNA in cancer. As you all probably know, cancer is a leading cause of morbidity and mortality, and over 23 million cases are expected annually by 2030. Up to 20% of those cases are hereditary, meaning that they are passed down from the parents. Up to 20% are familial, meaning that there's a lot of positives of the same type in the family, but they're not necessarily hereditary. They're caused by lifestyle or the environment. Up to 80% are somatic mutations. So those are mutations that occur after conception in any cell of the body, but not in the germ cells, so the sperm and the egg. Solid tumors are somatic mutations usually. Solid tumors are known to be very, very heterogeneous. And one of the markers for that could potentially be extrachromosomal DNA. So these extrachromosomal DNAs, or ecDNAs for short, are 1 to 3 megabase, usually circular double-stranded DNA. In these light microscopy pictures, you can see these blobs here, which are intact nuclei. This is a live cell, and all these bigger ones are the chromosomes. These smaller ones that you see, these circular ones, are the extrachromosomal DNA. They've only been found in cancer cells, and they're found in around 14 to 40% of solid tumors, and they seem to be associated with higher malignancy. There are several possible ways that these could form. It's not entirely clear, but it's been proposed that if you have two homologous repeats and they come close together, you could have misalignment, and the DNA replication machinery can slip, and you get formations of these structures. It can happen that things that are far apart in linear space, but close together in the three-dimensional space, therefore end up on the same ecDNAs, or ecDNAs of different, or several ecDNAs can be made in the same cell with this mechanism. The ecDNAs contain full oncogenes and regulatory regions, and they've proven to have impact in oncogene activation. So that offers a potential for diagnosis, prognosis, and therapeutics. Once these ecDNAs are generated, they're highly replicated, but they lack telomeres and centromeres, so they're unevenly segregated, meaning that it's really a coin toss whether they would end up in which cell after the cell division. That can lead to a lot of heterogeneity within the tumor. Also, the chromatin structure is very open, which can lead to high expression of the genes on there. As I mentioned, they usually have oncogenes, and basically a lot of different oncogenes have been seen, a lot of them in the MYC, c-MYC, MYC-N category, but also many others. So many of these oncogenes are targets for cancer treatment, and they're involved in the sensitivity and resistance of the cancer treatment. So it's important to detect these ecDNAs, and one way of doing that is by manual counting. So you can do that with staining and light microscopy. Now that seems to be quite laborious. It takes a lot of time. You don't know which molecules belongs to which cell after lysis, and you don't get any sequence information. Another way to look at them is with short read or medium read length NGS. Now with short reads, it can be hard to distinguish between structural variation, repeats, and retroviral integration that can happen in these ecDNAs. Also, it can be hard to see if the short read is coming from the ecDNA itself, or from the genomic DNA, or even from reintegrated ecDNA back into the genome. Also, there's no methylation information. So we decided we wanted to look at these ecDNAs with nanopore sequencing. Sergey Aganezov and the team found available data online. There's a team out of Berlin that already sequenced cancer cell lines that have ecDNAs, and they've made that data publicly available. So there are 3 samples that we're looking at. One is the CHP212 cell line that has the MYC and oncogene on the ecDNA, and we have around 4x coverage of that. Then we have the IMR 575 cell line, and we have around 2x coverage of that. And we have the Kelly cell line, and we have about 1.2x coverage of that. This data is roughly 2-3 years old, so we don't have too much data, but it's all sequenced on the MinION flow cell. Sergey then built a pipeline to be able to look at these ecDNAs. And what the pipeline does is it takes the ONT reads and maps them to the human reference. Then it calculates the coverage in 10 kb bins and identifies the outliers where there's higher coverage. And then you screen that against where you have outliers with higher coverage in normal samples, in normal genomic DNA samples. Then you take the tumor-specific outlier bins and do local alignments, and then you annotate them. So the first sample that we looked at was the CHP212. So as you can see in the alignment here, we have around the 4x that we knew that we have from the sample. And then we have 2 places where we have much higher coverage. These are both on chromosome 2, which is where the MYC-N gene is. After doing the de novo assembly, the result is this closed and fully resolved ecDNA circle at around 230x coverage. If we look at what genes are on this, we have the MYC-N as was advertised, and we have the MYC-N upstream transcripts. There's also a long non-coding RNA and some microRNAs. Then we do a refined representation, and we'll actually see that there are more regions on here than what we thought by just by looking at the alignment. So here, there are 6 big and then a few smaller regions that align back to the genome. The more orange in color, the further upstream they are on chromosome 2, and the lighter in color, the more downstream they are. So to make this even more clear, I've tried to color in down here on the alignments where these fragments come from. If we imagine doing a cut up here on the ecDNA and then flatten down all the different elements, then we can project it back onto the alignment, and you can see that the ecDNA is actually made up of these regions within the genome, but they're all in different order. The other sample that we looked at, and I'm just giving you all the data here now at the same time, is this IMR 575. So the first thing you'll see is in the alignment that we have mapping to lots of different regions with higher coverage. You'll also see that it's spread out on a bigger part of chromosome 2. We still have the MYC-N region, but we have almost all of the P arm of chromosome 2. The other thing you'll see is that we have different coverage, even in regions that are close to each other. So the result that comes out is this, from the bandage plot, is this one. So as you can see, it's not fully resolved, and it's not clear exactly what this looks like. So the reddish color up here has 107x coverage, and then the orange here has around double of that. And then you have others that are, the green is fairly close to that, and the turquoise is a bit higher as well. So what we think that this could be is either you have one circle that goes around here, goes down into the orange, sort of loops back onto the green and then goes in the orange again and then the blue, or something like that. So it's sort of an 8 structure. Or we could potentially have 2 ecDNAs that both have this orange bit in it. The genes again, we have the MYC-N upstream transcript, we have some microRNAs and some long non-coding RNAs. And again, you'll see that when we map back to the genome, we have 7 different parts. And the more green here is further upstream, and the more light green, the further downstream. Now this sample was sequenced at about a median reads of 10 kb and an N50 of 26 kb. And we do think that with either long reads or ultra long reads, we would be able to resolve this fully. I just wanted to step back and talk a little bit about how much coverage we actually need to be able to do these assemblies. So what I showed you before was all the data. It was this line down here. Now we're back to the CHP212 sample. And when we have 4x coverage of the human genome, we had around that 230x coverage of the ecDNAs, which was fully enough to do the assembly. We titrated that down, so 3x, 2x, 1x, and 0.25x of the human genome. And even at 0.2x of the human genome, we still have 14x of the ecDNA in this case. So if we do a dot plot of the titrated down samples against the 4x, or the full one, the full data set, sorry. We do have a dot plot that shows that we have complete assembly in all cases. So even a very shallow sequencing of the human genome, we still have fully resolved that ecDNA. And this opens up some possibilities about how you could use this type of sequencing. So with shallow sequencing of cancer samples and getting fully resolved ecDNAs, you could think about this being used as sort of in a targeted cancer sequencing setup, as a sort of a free add-on. Also, you could, with very, very short time on a MinION or a GridION, you could probably sequence enough coverage to get the ecDNA resolved within an hour, maybe 2. Or alternatively, you could do it very inexpensively on a flongle. So it's really only 1 gigabase of data at this 0.25x, which you could do on a flongle. Alternatively, you could multi-sample or multiplex and then sample multiple sites within a tumor to look for heterogeneity. So imagine that you took different samples from the same tumor and found that it was either the same ecDNA or different ecDNAs within that same tumor. And lastly, it wouldn't be a full nanopore presentation if I didn't talk quickly about methylation as well. As you all know, when you sequence whole genome sequencing with nanopore, you get methylation sort of for free. So we also looked at methylation at the single read level on all these 3 samples. And what you'll see is that there's not that big of a difference in the methylation around the MYC-N gene when you look at it at the high level. If you zoom in around the MYC-N oncogene, there is also not that much difference. But upstream of the MYC-N, you will see that the CHP212 sample has a significantly different methylation profile than the others. I'm not going to claim that I can tell you what that means, but it could potentially be part of this heterogeneity within the tumor if you have different methylation profiles within the same tumor. With that, that was my presentation on extrachromosomal DNA, and Phil James is now going to talk to you about a clinical assay that he's been working on. And let's take it away, Phil.
Hello, my name is Phil James, and today I'd like to talk to you about detecting germline variants in hereditary cancer using adaptive sampling. So to start off with, I'm just going to give a very brief overview of adaptive sampling and hereditary cancers, and then move on to show how adaptive sampling can be used on DNA from cell lines which have been derived from patients with hereditary cancer. So we've already seen this slide, but I'd like to draw your attention to this. Approximately 10% of cancers are hereditary. Now, a hereditary cancer is actually where a deleterious mutation in a cancer risk gene has been passed from the parents into offspring, and it's usually, or pretty much always, in a heterozygous form. Now, this forms the sort of basis of the 2-hit hypothesis of cancer, whereby every single cell in the body has already got 1 deleterious allele in a cancer risk gene, and it only takes a second mutation or second hit to potentially form a cancer. Now, just briefly, before we move on to the next slide, I'd like to say a little thing about structural variants. So this 10% value is likely to be somewhat underestimated, mainly due to people underestimating the impact of structural variants. And there's a really nice paper there if you're interested in that kind of thing. So previously I mentioned we're going to use adaptive sampling to look at this. And what actually is that? Well, it's a way of enriching or targeting for regions of a genome that you're interested in on the flow cell as you're sequencing, so you don't change the library prep or anything like that. And how this actually works is that the first few hundred bases of a strand is base called as it's translocating through a pore. And if it aligns and hits within a target region that you've predefined, it will allow it to pass through and continue sequencing. However, if it hits outside one of these regions, the read is rejected from the pore, freeing it up for a new strand to come in. If that happens, you then actually get a sort of enrichment for your target regions across your entire genome. Now, there's a few different ways that this can actually be implemented. However, the one that's been incorporated into MinKNOW comes from Matt Luce's lab. And Alexander Payne, who's the lead author on this paper, is actually speaking tomorrow. So if you're interested in adaptive sampling, definitely that's one not to miss. So as I said, this came out of Matt Luce's lab. And when it did first come out, it was called ReadFish. And I was really quite excited about this. So I did this sort of quite a silly experiment where I got some Na12878 DNA and I prepped the library, put it onto the flow cell. I asked the software on the left-hand side of the flow cell, I'd like you to select for BRCA1 and reject everything else. And on the right-hand side of the flow cell, I'd like you to select for BRCA2 and reject everything else. Then actually about halfway through, I decided swap the sides around. And this is what we can see on this graph here, the selection for BRCA1 and then the selection for BRCA2. Now this is, as I said, a little bit of a silly experiment, but it just showed how adaptive sampling sort of worked. However, what is actually useful, and in my opinion, that's actually large-scale research, large-scale panels of genes. And that's sort of where the hereditary cancer idea sort of came from. And I started on this sort of looking at something called Lynch syndrome, which is actually disruption to the mismatch repair genes. So this sort of disruption can happen in a number of different ways. It can be like single nucleotide polymorphisms, copy number variants, or even hypermethylation of the promoters. But the first job was to actually find out, what do I want to target? And sort of like any good scientist, I decided that I'd Google it. So I did, and there's a number of companies, a number of research panels and papers which are out there, which all pointed me towards this sort of cluster or handful of genes. So that was that. I took these genes, I went into IGV, and I decided, I took the coordinates and put them into a targets file, and it was sort of like, job done. However, something caught my eye, and that was an expanded Lynch syndrome panel. And I was like, oh, well, there's no problem, I'll just add all of these ones in. And then I noticed that you can actually have specific hereditary cancer panels, and these look at things that, like breast cancer, ovarian cancer, prostate cancer. And I just thought, why not? We've got all of these genes, we can put them in, it's no problem. And then things got a little bit silly, because obviously these expanded hereditary cancer panels, or even these absolutely massive research panels. Now, the point here is that I'm not an oncologist. I don't recognize most of these genes, and I certainly don't know what they do or how they relate to cancer. But the point is that somebody does. And it was no more effort for me to actually put all of these genes into a targets file than it was for me to do the original Lynch syndrome panel. And that's the sort of thought that struck me on some idle Thursday afternoon, that I've created this theoretical, absolutely massive panel, which, if I was trying to do this during sort of using traditional methods like PCR or pull downs, that could take me months or years. The point was that at the moment it was theoretical, so I had to try this on something. So I went to my freezer, and I found I had a box of hereditary cancer samples, as one does. And I chose 4 at random. And these turned out to be ovarian cancer, and 2 breast cancer samples with a family history of breast cancer, and 1 which had a BRCA-negative breast cancer sample. Now, BRCA-negative means that there's no variants have been found in BRCA1 or BRCA2 by sort of the external validation of these samples. So I took these and HG001 as our non-cancer control and sequenced them for 72 hours on a GridION, 1 per flow cell, and I did 2 nuclease flushes. And we got over 22.5 gig per sample under this adaptive sampling conditions, which was really great. I thought this is wonderful, but we need to see how this actually relates to our on-target rates. So this next graph, I really like this graph mainly because it moves, but each one of those bars at the top is one of our targets. And it's an hour-by-hour demonstration of the read depth that's been calculated for these. And we see that we can actually get 57x across all of our targets, which was, I thought, quite phenomenal, because I was only aiming for about 30x. And we can see in the bottom left and the bottom right the per base coverage of BRCA1 and BRCA2. And here I'd just like to point out that we're covering the exons, but we're also covering the introns. We've got the whole gene there and some of this padding around the outsides. In the middle, we've got the non-targeted regions, which are populated by the 400 or 500 base pair rejected reads, and it goes somewhere around 4 to 5x there. So the next job was to go, okay, do we have any pathogenic variants in these genes? Can we find them? Now, I'm not going to go through all of the bioinformatics here, but basically what we can see on the right is all of the SNPs that have been detected, which have been intersected with the ClinVar database, and all of those which have been deemed as pathogenic. And we can see that in all 4 of our cancer samples, we find 1 SNP, and in NA12878 we don't find any. And now this is particularly interesting from NA14623, which was our BRCA-negative breast cancer sample. So I'd just like to quickly go through some of these, because I find the sort of mechanisms quite interesting here. But one of our first samples is a cysteine to glycine missense mutation in exon 5, and this results in a functionally null protein. So the protein's made, but it just doesn't do what it's supposed to do. This next one is a different type of mutation. It's a nonsense mutation in exon 13, and this basically incorporates a stop codon, so you get a truncated protein. The one I find the most interesting, though, is this mutation in an intron. And what actually happens here is it moves the splice acceptor site back a few base pairs and forms a cryptic splice site. So actually in your mRNA, you have a 59 base pair insertion, which also includes a stop codon, and you get a truncated protein again. And now this just, I think, demonstrates why you should sequence your introns quite nicely. However, we spoke about our BRCA-negative sample, and we still found a pathogenic SNP, or a SNP that was designated as pathogenic in the ClinVar database. Now, this is in a gene called PALB2. Now, what this actually does is it forms a complex with BRCA1 and BRCA2 and a 4th gene, which is RET1, I think. And this forms a function of homologous recombination repair. Now, disruption to this complex obviously has quite negative effects on that complex formation. And it's mostly studied the mutations and this interaction with this complex from the perspective of BRCA1 and BRCA2 and the mutations which disrupt these cold-coal interactions. PALB2, however, in BRCA-negative samples has been really proposed as a good sort of candidate gene for looking for mutations due to the potential disruption of this complex. However, this is an intronic variant, and we don't know what this actually does, so we need to look at this sample in a little bit more detail. But before we do that, I'd quite like to go on and talk a little bit about BRCA1 and 2 themselves. Now, we've been talking about homozygous or heterozygous SNPs specifically, and I'd like to show just in a sort of concept of phasing, because with heterozygous SNPs in sort of normal or in genetic hereditary conditions, it's quite useful to know which haplotype your heterozygous variant is on, because sometimes you can have one on one haplotype and another on another haplotype, and that's called compound heterozygosity. And a sort of classic example of that is cystic fibrosis in the CFTR gene, and that's where read phasing really comes into its own. Here we can also see that we can phase our reads completely across BRCA1 and BRCA2. Now, these are reasonably complex genes. So not only does phasing allow us to look at haplotypes sort of resolved methylation calls, structural variant calls, and SNP calls, for example, but in the case of BRCA1 and BRCA2, we know that they've got some pretty nasty pseudogenes associated with them. And the fact that we phase into 2 haplotypes and not 4 means that we're probably not having any problems with these extra pseudogenes. But more than that, we can actually sort of double down on this and go, actually, our mapping qualities here, not over 97% of our reads are of a mapping quality of 60, meaning they're uniquely mapped to BRCA1 and BRCA2 as in not having a problem with these pseudogenes. So far, we've got our SNPs, we've got some phasing, we've found some interesting things, but what else can we actually do with this data? As I mentioned before, we've got some padding regions on the end, and we're using the native DNA. So theoretically, we could look at methylation in our promoter regions. However, why would we want to actually limit ourselves to that? What we could actually do is take 15,000 promoter regions from the eukaryotic promoter database, which have more than 4 CpGs in them, and these are experimentally validated promoter regions, and just put those into our targets file. And that's actually what we did. We can see here that 4 out of the 5 samples, we actually get 95% of these regions covered to more than 20x, which is perfectly fine for methylation calling. One of them didn't work quite so well, but you sort of got to show it all here. We also actually have NA12878 in here, and we've got bisulfide data for this as well, so we can compare the two and see how they match up with each other. And actually what we see that over 2.8 million CpG sites, we get a nice correlation coefficient of 0.958, which is really, really good. And it's also worth bearing in mind that we're sequencing the native DNA, so we don't have any real problems with PCR bias or GC bias or things like that. Now, this is just an example of the per CpG methylation calling across all of our samples, just for one gene in particular. I thought this was a nice example. And this is the MSH2 gene, which was one of the core Lynch genes, the mismatch repair genes involved with Lynch syndrome that I was speaking about earlier. Now, while we've got lots of differential methylation in different regions of the genome between the samples, this was a really nice example where all of our cancer samples had hypermethylation in this promoter enhancer region for MSH2, whereas NA12878 didn't. Now, we weren't really expecting to see this, and I'm not suggesting that these are Lynch syndrome sort of samples, but I just found that it was quite an interesting concept. And hypermethylation is actually usually associated with MLH1, not MSH2, but still, it was a stark contrast and a very good demonstration of per CpG methylation calling. Now, we're sort of moving into the sort of end of the presentation here, and there's one last thing I'd like to talk about, and that is based on our throughput, we have about 22.5 gig of data or more of each sample. And we've got all of our on-target reads, we've looked at SNPs, we've looked at methylation, stuff like that. But what can we do with those 4 500 base pair rejected reads? Now, we can align them to the genome, and we can actually get sort of low-pass whole genome sequencing, look at our genome using these short reads. Now, the thing here is that when people talk about low-pass whole genome sequencing, they're sort of talking about less than 1x, and you can sort of look for copy number variants which are at the megabase scale or even chromosome scale in the case of trisomy 21. But here we've got between 4 and 5x, so what can we actually find with this in terms of copy number variants? So again, I'd like to go back to the BRCA negative sample, and I find this bit quite phenomenal. We find a 60 kb copy number variant deletion, which encompasses a gene called PIP. Now, PIP, or the prolactin-inducible protein, is actually quite strongly linked to anti-tumor immune response. And I find the fact that we can find a germline variant, which encompasses this cancer gene, quite phenomenal in this hereditary cancer sample. And furthermore, what we can actually do is we can use our alignments, and we can link our primary and our supplementary alignments, and we can actually find the exact breakpoints of this copy number variant. And when we actually look in the literature, this copy number variant has been previously found, and it's called the Sao Paulo PIP copy number variant. Now, I just have to say that actually PIP is usually looked at from a somatic point of view, and sort of expression rates of PIP seem to be linked with better prognosis and association with chemotherapy. So we don't actually know what the impact of a copy number variant in the germline actually is, but it's certainly interesting. So just to finish off, we've just got a nice summary slide here. Going back to Sissel's presentation, long reads can identify oncogenes in these quite fascinating circular extrachromosomal DNA molecules. Furthermore, that these can actually be assembled, and you can generate methylation profiles from these. In this half of the talk, we were talking about adaptive sampling and how these panels could be quickly and easily designed and to give us quite high coverage of our target regions, which allow us to identify things like pathogenic variants. We can look at methylation and study compound heterozygosity, and also we can look at phasing and study compound heterozygosity and identify hypermethylation in promoter regions and, for example, CPG islands. Furthermore, we can use those rejected reads to look at copy number variants and locate breakpoints by linking supplementary alignments. So as a very last thing, I'd just like to go right way back to the start, which is the talk title, which is something we always like to explain, and that was For Want of a Nail. Now this comes from a really old sort of proverb, which basically highlights that small aberrations in a system can actually have really large deleterious effects later on in life. And I sort of thought that sort of encompassed the sort of ethos of this talk quite well. But secondly, I also thought that it always shows that there's this requirement for reasonably simple tools to try to help look and examine rather complex problems. So with that, I'd just like to say sort of whatever research you're actually doing and whatever tools you are using, hopefully we've shown that Nanopore can sort of fit quite well into that arsenal. And hopefully in the future, not too far away, no one will want for a nail. Thank you for your time.